So we are going to see isolation of bacteriophages. First of all, from where to take the sample? The sample of bacteriophage isolation will be taken from the sewage, and sewage, you know that there are so many gram-negative short-rolled sewage. They contain their specific bacteriophages. So that is why we are taking sewage sample in the form of our crude bacteriophage sample. Now, after that, we will try to understand that what is the host culture we have to take. Here we are taking floor culture of Escherichia coli. Why? Because we know that Escherichia coli contains so many bacteriophages which are already present in the sewage sample. So there may be more probability of getting a specific type of bacteriophages from the sewage sample, and that is why we are taking sewage sample and respective host culture, that is Escherichia coli. Now, after collecting the sample, we need to pre-treat the sample because directly we are not going to isolate the bacteriophages. So there are two methods. Either we can go for filtration or we can go for centrifugation. According to our lab requirement, we will try to carry out centrifugation at a specific RPM in 4 to 5 minutes, and then after we will collect the supernatant, that is the upper portion of the test tube in which bacteriophages will be there. Now, why they go to upper phase? Because they are very light in the weight, so they will go to the upper part, that is called supernatant, and we are going to take now topagar and base agar for our isolation. Okay? Now, this topagar when it is hot at that time, we need to add 0.1 ml of supernatant, which will be our crude phage sample. This is considered as crude phage sample. Now, crude phage sample will be added in the topagar tube in the quantity of 0.1 ml exactly, and then after we add our floor culture of Escherichia coli, which is young and actively growing, and the quantity will be added, that will be 1 ml. Now after that we will mix this, rotate this topagar tube nicely, and then after we pour it on the base agar plate. Now after pouring, we will allow it to solidify. After solidification, we will give proper incubation, that is 24 hours at 37 degrees centigrade temperature in incubator, and in the next day we are going to see the this media plate, and we will come to notice that there is lone growth of bacteria. Means bacteria are grown in entire plate and there is no other place where bacteria are not found. But in the among this particular loan, there will be some clear area which will be very smaller one. It will be clear and small. It may be hazy and bigger, depending on the type of the bacteriophage. Okay, so we will see this clear area which are just like the incubation zone we have seen in the antibiotic sensitivity of bacteria. And now these clear zones are nothing but they are bacteriophages, and they are the growth of bacteriophages. Now one question will come in our mind that why bacteriophages are there and cannot be seen. Now the reason is that because bacteriophages are ultramicroscopic, they cannot be seen by naked eye unless and until you utilize the electron microscopy. So that is why even if they are here, they have eaten the bacteria and they have grown in the form of colony, but that colony cannot be visible by our naked eye. So it will appear in the form of clear small area. Now these clear small area are given a specific name, that is called plaque. So now we are going to observe the plaque, and then after we will conclude our result that we can isolate the bacteriophages from the sewage sample in the form of the plaques with the help of Escherichia coli host culture. Thank you.